Welcome to Chapter 11, The Instructions for Life, DNA and RNA. Until the start of the 20th century, scientists knew very little about DNA. It took years for investigators to conclude Mendel's factors, which were genes, were located on chromosomes. And there was a controversy over whether DNA or protein was the genetic message. Experiments using viruses showed only DNA directed the formation of new viruses. Alfred Hershey and Martha Chase determined that DNA is the genetic material. Their experiment involved a virus that infects bacteria such as E. coli. This is called a bacteriophage virus. They wanted to know which part of the virus entered the bacterium. The capsid of the virus is made of protein, and the DNA is inside the capsid. Radioactive tracers showed that DNA, not protein, enters the bacterium and guides the formation of new viruses as the genetic information instruction manual. Therefore, DNA must be the genetic material. So here we see the anatomy of a virus, and we can see how the virus has a protein capsid that encapsulates or contains the DNA. And the virus, called the bacteriophage virus, infects bacteria by injecting its DNA into the bacteria, and the DNA is going to use the bacteria's cellular machinery in order to create new viruses. Here's an actual photo showing you the virus and the bacterium. There was also a time when the structure of DNA was not known. There was a race to determine the structure of DNA. Chargaff's rules states that DNA contains 4 types of nucleotides, and each of these nucleotides pair up with a complementary nucleotide. The amount of A, T, G, and C in DNA varies from species to species. A matches with T and G matches with C, so the amount of A nucleotides equals T and G equals C. All nucleotides contain phosphate, a 5-carbon sugar, and a nitrogen-containing base. Here we see a nucleotide with the 3 parts that it is made of. We have the phosphate group, the sugar, and the nitrogen base, which is going to determine whether you have the letter C, G, A, or T. Here is the composition of DNA and RNA. Both DNA and RNA have phosphate. However, DNA and RNA are going to be different in what sugar that they contain for their nucleotides. As you can see here, DNA is going to have the sugar called deoxyribose, whereas RNA is going to have the sugar called ribose. The only difference in terms of structure between these 2 sugars is the H versus the OH on the second carbon. Furthermore, another difference between DNA and RNA is the nitrogen bases. For RNA, instead of having a T, which stands for thymine, RNA has a U instead, which stands for uracil. Franklin's X-ray diffraction data. Rosalind Franklin was studying the structure of DNA. Her data showed DNA to be a helix with some portions repeating over and over. Her discovery was monumental into eventually discovering the full scope of the structure of DNA. Here we have a slide going over X-ray diffraction technology. In 1951, James Watson and Francis Crick set out to bring together all the data on DNA and build a model. The model suggested how DNA replication works. And we remember that replication is the process of duplicating or making another copy of the DNA. Their model holds true today with few changes, and eventually Watson and Crick won the Nobel Prize. DNA is a double helix, like a twisted ladder. Deoxyribose sugar and phosphate molecules are bonded. They form the backbone or the sides of the DNA, with the bases making up the rungs of the ladder, being in the middle. A is going to match with T nucleotides, and G is going to match with C nucleotides. Hydrogen bonding between the bases, AT and GC, holds the halves of the helix together. So here we see the structure of DNA. We have the sugar in green and the phosphates in yellow. They are going to make the sides of DNA. While in the middle, we have the nitrogen bases complementarily matching each other. G matching with C, A matching with T. If we zoom in, we can see how the nitrogen bases of each nucleotide are going to bond through hydrogen bonds. Notice how A and T are going to bond together through 2 hydrogen bonds. However, if we look at G and C, we can see that they bond through 3 hydrogen bonds. Replication of DNA. Replication is the process of copying DNA before cell division. We remember that DNA replication occurs during interphase and specifically in the S phase of interphase. In the process of DNA replication, the 2 strands of DNA are first separated, and each strand is going to serve as a template for forming the new strand. DNA replication is semi-conservative. Each new DNA molecule is made of 1 parent strand and 1 new strand, instead of conserving both parent strands. Replication requires the unwinding of the DNA, 2 strands, by helicase. Replication requires complementary base pairing, and also replication requires rejoining or joining the new strand to the original parent strand. And this happens through DNA polymerase and DNA ligase. New DNA molecule is exactly identical to the original molecule. However, it conserved only half of the or one of the parent strands for each new DNA molecule. Again, semi-conservative replication is going to only result in the conservation of 1 parent strand per new DNA molecule. To show a simplistic diagram here, we have the original parent strand with 2 strands of DNA and 2 parent strands, but after DNA replication, well, first we need to separate those parent strands, and then during DNA replication, DNA polymerase is going to use those 2 parent strands as templates and add the new daughter strands. And finally, we have our 2 new DNA molecules with 1 parent strand and 1 new daughter strand. So as you can see, the 2 new DNA molecules, they only have 1 parent strand from the original 2 parent strands. So this is why replication is called semi-conservative. Here we see another diagram where we have 2 strands from the parent strand first unwinding through the help of helicase and the formation of the green daughter strands, resulting in eventually two new DNA molecules with conserving one parent strand each. In eukaryotes, DNA replication begins at numerous origins of replication. These origins of replications are going to form replication bubbles, and the bubbles are going to spread in both directions until they meet. So here we see eukaryotic replication. Helicase is going to unwind the twisted DNA into two straight parent strands, and DNA polymerase, as we can see here, are going to form the daughter strands. Now let's take a look at RNA structure and function. RNA structure and function. RNA is called ribonucleic acid. And as we saw earlier, RNA contains the sugar ribose instead of the sugar deoxyribose, which is contained in DNA. Also, RNA is not going to include a thymine base. RNA includes a uracil base instead. And also RNA is single-stranded, not double-stranded like DNA. There are three major types of RNA. We have messenger RNA, also called mRNA, transfer RNA, which is also called tRNA, and ribosomal RNA, which is called rRNA. And here we see the structure of RNA. And just like DNA, RNA is also going to have the phosphate and the sugar backbone, which is the side of the RNA. And in the middle, it's going to be the nitrogen bases. But instead of T in RNA, we have U. So here's a comparison chart going over the similarities between DNA and RNA and the differences. There are three types of RNA. We have mRNA called messenger RNA, tRNA called transfer RNA, and rRNA called ribosomal RNA. Messenger RNA is important because it is going to carry the genetic message or information to the ribosomes to make a protein. Transfer RNA is important because it transfers amino acids to the ribosomes, and we remember that amino acids are the building blocks of proteins. Each type carries only one type of amino acid. rRNA, called ribosomal RNA, is important because rRNA is going to help build the ribosome, or it is going to form a part of the ribosome. The rRNA is going to be produced in the nucleolus, which is inside of the nucleus. In the early 1900s, Sir Archibald Garrett suggests a relationship between inheritance and metabolic diseases. He was the first to suggest a link between genes and proteins. DNA provides a blueprint to synthesize proteins. Information is going to flow beginning with DNA, and then to RNA, and then to protein. We call this process gene expression. Gene expression is going to be divided into two parts. The first part of gene expression is the transcription of the gene from a DNA to an mRNA. The second part is called translation, where the mRNA is going to be used in order to build our protein. The rRNA and the tRNA are going to assist in the process of building the protein. Here we see the flow of genetic information starting from DNA all the way to a protein. So as we can see, we start with DNA, and through the process of transcription, we are going to convert DNA into mRNA. Notice in the diagram that DNA and mRNA are going to start off inside of the nucleus. The process of converting or creating mRNA from DNA is called transcription. Where we transcribe the DNA to the mRNA. Next, the mRNA has to exit the nucleus and go into the cytoplasm through the nuclear pore, which is a hole in the nuclear membrane. mRNA is going to be converted or used to make a polypeptide or a protein, which is also called the polypeptide. Okay. This process is called translation. The genetic code is going to produce the protein by using the information that is stored in the mRNA and originally from the DNA. Translation is going to produce a unique amino acid sequence depending on the sequence of the mRNA. mRNA is going to be read in groups of 3 nucleotides, which are called codons. Groups of 3 nucleotides on the DNA is called a triplet. And a codon is going to provide the information for a very specific amino acid. Also, we have start and stop codons, which start and stop translation. So remember, the DNA is going to code for the mRNA, and the mRNA is going to code for the amino acid. Here are the various codons in groups of 3 from mRNA, and each codon is going to match with a specific amino acid. So as you can see here, the codon UUU is going to code for the amino acid phenylalanine. Another example is UAC is going to code for the amino acid tyrosine. And in order to start the translation process, we need to have a start codon, which codes for the starting amino acid methionine. The stop codon is going to be UAA, UAG, or UGA. During transcription, complementary RNA is made from a DNA template. A portion of the DNA unwinds and unzips at the point of attachment of RNA polymerase. RNA polymerase is going to create the new RNA strand from the DNA. Bases join in the order dictated by the sequence of bases in the template DNA strand. So firstly, we need to separate the 2 DNA strands. That happens through helicase, and then the template DNA strand is going to be used to create our new RNA strand. in green. So we only use one of the DNA strands called the template DNA strand. Also, RNA polymerase is the enzyme that's going to help create that new RNA molecule. After RNA is transcribed from DNA, RNA must be processed before it can exit the nucleus into the cytoplasm. The newly made pre-mRNA is what the unprocessed mRNA is called. Processing includes capping and adding a poly A tail, which provides stability for the mRNA when it enters the cytoplasm from the nucleus. Also, non-coding regions of the mRNA are removed. These non-coding regions are called introns. The coding regions are called the exons. Coding means that it is going to code for a certain protein. Non-coding means that it does not code for a protein. Alternative splicing can produce different versions of mRNA, leading to different proteins. So certain parts are cut out and certain parts are left in different combinations. After the pre-mRNA is processed, the now mature mRNA can leave the nucleus and enter the cytoplasm, where it is going to interact with a ribosome. Here is a diagram of mRNA processing. The first thing which occurs is the addition of the 5' cap and the poly A tail on the ends of the pre-mRNA. This is going to ensure stability for the mRNA in order to survive in the cytoplasm. Next, the introns, which are the non-coding regions, are going to be cut out, and the remaining exons are going to be spliced together, glued together. Now we have our mature mRNA ready to leave the nucleus and enter the cytoplasm, being able to survive. Here is the structure of tRNA, which is important for transferring amino acids to the ribosome during translation. tRNA has one end called the anticodon, which matches with the respective codon on the mRNA. And on the other end of the tRNA, we have the attachment site for the specific amino acid. Here's a more simplistic picture of a tRNA. We have the amino acid attached to the tRNA, and we have the anticodons on the other end. In translation, tRNA is going to bring a specific amino acid. Each tRNA is unique in that they have a different sequence of anticodons. Anticodons are going to be 3 nucleotides that are going to pair up with the codon on the mRNA. After translation is complete, a protein contains the sequence of amino acids originally specified in the DNA. And so we remember that amino acids are the building blocks of protein. And so this all originally was coded in the DNA. Ribosomes are composed of protein and rRNA. The ribosomes are often called the protein factory. This is the site of translation where protein synthesis is going to occur. At the ribosomes, the mRNA binds to the ribosome, and 2 tRNA molecules will bind to the ribosome during translation. There's 3 attachment sites on the ribosome. 2 of them are called the P site, where the tRNA is attached to a peptide, which is the chain of amino acids, and we have the A site for the newly arrived tRNA with an amino acid. The 3 steps or phases of translation are initiation, elongation, and termination. Let's look at each phase in detail. Initiation is when the mRNA first binds to the small subunit of the ribosome. Next, the large subunit of the ribosome then joins and combines with the small subunit. Elongation is when the polypeptide is going to be lengthened 1 amino acid at a time. Termination is when one of 3 stop codons is reached, and the stop codon is going to code for a release factor, which causes the disassembly of the mRNA from the ribosomal subunits. The polypeptide is then released. Here's the diagram of initiation, where the mRNA is first going to bind to the small subunit of the ribosome. Afterwards, the large subunit is going to bind to the small subunit in order to complete the ribosome, as we see here. Notice also that the initiator tRNA with the complementary anticodon to the start codon is going to bind to the mRNA with our start amino acid methionine. And also notice after the large subunit binds to the small subunit, the initiator tRNA is at the P site. In elongation, the amino acid chain or the peptide chain right here is going to grow longer. As the mRNA is being read by the ribosome, the respective amino acids are going to be added to the chain. So we see here the tRNA with the growing polypeptide chain is going to be in the P site, while the new tRNA is going to enter the A site of the ribosome. The anticodon is going to match with the complementary codon sequence and bring in the unique amino acid to add to the growing polypeptide chain. So if we look at step 2, after the new tRNA comes in, the peptide chain is going to transfer to the new amino acid. And as we can see, that creates a longer amino acid here. The tRNA in the P site no longer has anything attached to it, and therefore it's kicked out when it reaches the E site, the exit site. So how did it move from the P site to the exit site? The ribosome actually moved forward. So the ribosome moved forward, and that caused a shift of the mRNA and also the tRNA. Now the tRNA in the A site is going to move to the P site, as we can see here. And the new tRNA is going to come in, and the cycle will continue. And in our last step of translation, when we reach the stop codon on the mRNA, a release factor is going to enter the A site, which is going to cause the release of the polypeptide and also the disassembling of the ribosome, the mRNA, and the tRNA. The polypeptide is going to now travel to the endoplasmic reticulum, where it will be further processed. So here's a summary of gene expression in eukaryotes. Everything starts in the nucleus, and in the nucleus, the DNA is going to be transcribed into the mRNA. The pre-mRNA has to be processed, and then the mature mRNA exits the nucleus through the nuclear pore, and the mRNA, now in the cytoplasm, travels to the ribosome, where it will be translated eventually into a peptide chain or amino acid chain. Eventually, when we reach the stop codon, the peptide is going to be released and travel to the rough endoplasmic reticulum, where it's going to be further modified and folded to produce a protein. Gene regulation. The body contains many cells that differ in structure and function. Only certain genes are active in cells that perform specialized functions. This is why we have different types of cells. However, housekeeping genes are going to always be on because they govern functions common to all cells. The activity of selected genes accounts for specialization. So if you look at the diagram to your right, you have red blood cells, muscle cells, and pancreatic cells. All three cells are going to have housekeeping genes that are on, but only red blood cells have the hemoglobin gene turned on, which is the primary protein in red blood cells that carries oxygen. For pancreatic cells, the gene for insulin is going to be turned on. And for muscle cells, the gene for the protein myosin is going to be turned on. All the other genes will be turned off for genes that are not needed in those specialized cells. So red blood cells will not produce insulin or myosin, and muscle cells will not produce hemoglobin or insulin, and pancreatic cells will not produce hemoglobin and myosin. Even though all cells have the same genes, certain genes are turned on and off, and this is going to make the cells unique or specialized. Now let's take a look at gene expression in prokaryotes and how they differ from eukaryotes. For example, E. coli lives in our intestine and can quickly adjust its enzymes according to what we eat. If we drink milk, E. coli immediately begins to make 3 enzymes needed to metabolize lactose, which is the sugar inside of milk. An operon is a cluster of bacterial genes along with DNA control sequence. Lactose is not available most of the time, so E. coli normally does not transcribe the genes of the operon. When lactose is not present, repressor binds to the operator and RNA polymerase cannot attach to the promoter. This is going to inhibit or stop transcription, which is ultimately not going to produce the proteins necessary to metabolize lactose. So here we see the lac operon, which is a series of genes, and also the promoter and operator and the regulatory gene. So if we do not have lactose, or if the E. coli does not have lactose in the environment, a repressor is going to bind to the operator, and this prevents the RNA polymerase from binding to the promoter in order to transcribe the lactose metabolizing genes. So if lactose is present in the environment and the bacteria wants to use the lactose as an energy source, the lactose is able to remove the repressor from the operator, inactivating the repressor, which allows the RNA polymerase to bind to the promoter, and transcribe the genes necessary for lactose metabolism. When lactose is present, again, it binds to the repressor, inactivating the repressor, which cannot attach to the operator, and the RNA polymerase is able to bind to the promoter, and transcription is going to occur for the genes that are going to be responsible for making the enzyme that's going to digest the lactose, which is lactase. Another example is the binding of tryptophan. A gene for synthesis is normally on. However, if there is an excess of tryptophan, it causes the operator to be turned off with the same mechanism. Let's take a look at gene expression in eukaryotes. In eukaryotes, each gene has its own promoter, and there are a variety of mechanisms employed, and these mechanisms affect whether the gene is expressed or if it's not, the speed of how fast it's expressed, and the length of time for the expression. So remember, gene expression is when the gene is read as DNA, and that information is used to create a protein. Some mechanisms occur in the nucleus and others in the cytoplasm. In the nucleus, the mechanisms include chromatin condensation, mRNA transcription, and mRNA processing. And in the cytoplasm, delay of transcription and the duration of mRNA or protein. So here we see the different levels where gene expression can be controlled within the nucleus, either at the chromatin condensation level, at the DNA transcription level, or the mRNA processing level. For example, in chromatin condensation, the more tightly the DNA is wrapped around the green proteins here, called the histones, the less that the DNA is going to be read or expressed. And if we look at the DNA transcription level, we need the RNA or the mRNA transcript from DNA in order to make eventually the protein. So if transcription or the rate of transcription is lowered, then that means that gene expression is also going to be lowered due to the decrease in the production of the RNA transcript. So here's gene expression in the translation level and protein activity, and also the cytoplasm. Translation is going to be the last step of gene expression, where we're going to create the protein from the mRNA transcript. And so when we create a polypeptide, also gene expression can be controlled in the cytoplasm, where the protein can actually be degraded, or it can survive as a functional protein. Again, we will revisit chromatin condensation. The more that chromatin is condensed, meaning the more the DNA is wrapped around the proteins, this is a way to keep the genes turned off. The more tightly the DNA is wrapped around the proteins, the less gene expression is going to occur. Heterochromatin is the dark staining regions of tightly compacted inactive chromatin. The bar body is a second X chromosome in mammalian females, which is inactivated. So an example of X inactivation, or the presence of the bar body, is in female tortoiseshell cats. So as we know, females have two X chromosomes. So here we have the two X chromosomes, And for this female kitten, she has one allele for the orange color and one allele for the black color. However, early on in development, in the development of the embryo, some of the cells are going to have one of these X chromosomes inactivated. So we're going to have two populations of cells that either have an inactive X chromosome for the black color, and we have another population of cells which have the X chromosome for the orange color inactivated. This results in a kitten that shows a tortoiseshell color, meaning that the kitten has both black colored fur and areas of orange colored fur, representing the cells early on that were black and orange due to X inactivation during early development of the animal. Euchromatin is the opposite of heterochromatin. Euchromatin is when we unpack the heterochromatin, and therefore we have the active genes. A nucleosome is a portion of DNA wrapped around histones. And when we group together nucleosomes together, we form chromatin. A transcription activator pushes aside histones so that transcription can begin. So if I kind of draw out the order of DNA being wrapped around, DNA is going to start, and next we have DNA wrapped around the histones, which is the nucleosome. And when the nucleosomes group together, they form the chromatin. And when the chromatin group together or pack together, they form the chromosome. So this is kind of the level of organization from the DNA to the chromosome. So here we see the nucleosomes, which is just the DNA in blue wrapped around those purple proteins called histones. And when the nucleosomes group together, we call that a chromatin. As we can see here, there is a chromatin remodeling complex that interacts with the chromatin in order to kind of unwind that DNA and make it more loose so that it's able to be transcribed more easily. DNA transcription in eukaryotes. So we have the same principles as prokaryotic transcription, but with more regulatory proteins per gene. This allows for greater control, but also a greater chance for malfunction. The first term we need to know is transcription factors. Transcription factors are DNA binding proteins that help RNA polymerase to bind to a promoter. So the job of transcription factors is to help RNA polymerase bind to our starting promoter, which is going to start transcription. Several are needed in each case, and we need all of them. To form the complex means that, or after we form the complex, it helps to pull apart the helix and helps to position the RNA polymerase. The same ones are used in different combinations. If one transcription factor is defective, it can have serious effects, such as Huntington's disease. We can also, or transcription factors speed up transcription and also bind to the enhancer region of DNA. Okay, so here we see the different transcription factors. and RNA polymerase. So before RNA polymerase binds to the promoter, where transcription is going to start, the transcription factors here are going to form a complex here, combined together. And also we have another transcription factor called the activator bound to the enhancer region of DNA. So the transcription factors, they're going to help RNA polymerase to bind to the promoter. And once it does, the transcription factors are going to form a complex here and also kind of bend the DNA, causing a hairpin loop to result, which is going to allow the initiation of transcription. It is possible for a single transcription factor to have dramatic effects on gene expression. MyoD alone can activate the genes necessary for fibroblasts to become muscle cells. So MyoD is a transcription factor which can convert the fibroblast cells to differentiate into muscle cells. EY transcription factors can bring about the formation of a complete eye in flies. So transcription factors can have huge effects with development. Next, we will discuss mRNA processing. After transcription, introns must be removed and exons spliced together. Introns are the non-coding regions of the mRNA, and exons are the coding regions of mRNA. So what does coding mean? Coding means that we can produce a protein. So that portion of mRNA produces a protein, or it's going to be translated into a protein. Non-coding means no protein produced. Alternative mRNA processing allows cells to produce multiple proteins from the same gene by changing the way exons are joined. The fruit fly gene DSCAM can produce over 38,000 different combinations, depending on alternative mRNA processing. So here we see two different types of proteins that are produced from the same mRNA transcript. But how can the same mRNA transcript produce two different proteins? And this is due to RNA processing and RNA splicing in different ways. So in the first transcript, we splice out the introns and we splice together the exons in the order A, B, C, D, E. And this unique sequence of the mRNA is going to produce a unique protein, product 1. In the second scenario here, we have the same mRNA transcript, but we also splice out the introns, but we have a different splicing together of the exons, a different sequence. Instead of A, B, C, D, E, we have A, B, D, E, and this sequence is going to produce a different protein product. Next, we have the control of gene expression at the translation level. So in the cytoplasm, it contains proteins that determine whether translation takes place or not. Environmental conditions can delay translation. For example, in red blood cells, if we don't have a heme group, hemoglobin is not able to be produced. The longer the mRNA remains in the cytoplasm before it is broken down, the more gene product is produced. But how does the mRNA last longer in the cytoplasm? This is affected by the length of the poly-A tail. The poly-A tail is added to the end of the mRNA during mRNA processing and allows the mRNA to last longer in the cytoplasm. And also the presence of some hormones allows the mRNA to last longer in the cytoplasm. So these factors at the translational level is going to affect gene expression. Next, we will discuss protein activity. Some proteins are not active immediately after synthesis. For example, insulin must be processed before it is in active form. If we look at the example in the diagram, this polypeptide is inactive, but in order to activate it, we must cut it into smaller polypeptides to have an active polypeptide. This allows protein's activity to be delayed until needed. Signaling between cells in eukaryotes involves constant sending of chemical signals that influence the behavior of other cells. During development, signals determine what a cell becomes. Later, they help coordinate growth and daily functions. So the development of a cell into a certain type of a cell is called cell differentiation. So how do we know if a stem cell is going to become a muscle cell, a brain cell, a bone cell, or etc.? This happens through signaling, cell signaling. And also the growth of an organism and the functions of an organism is determined by cell signaling. The cell signaling pathway begins when the chemical signal binds to a receptor. So the chemical signal molecule binds to a receptor. And that receptor is on our cell called the target cell because we're trying to bind that chemical signal to that receptor. The receptor is located on the plasma membrane of the target cell. And we remember that the plasma membrane is the same thing as a cell membrane. After binding to the receptor, this initiates a signal transduction pathway inside of the cell, producing a cellular response. The end product affects the cell, not the original signal itself. So here we see a diagram of cell signaling. We have the receptor here in blue and the chemical signal here, the molecule in green. The chemical signal molecule is going to bind to the receptor on the plasma membrane of the target cell. And this produces a signal transduction pathway. Signal transduction means the propagation or the traveling of the signal. So signal transduction is the sending or traveling of the signal along a certain pathway. This is going to end up activating, in this case, a transcription factor. And the transcription factor, as we saw earlier, is going to bind to an enhancer region on the DNA, and it's going to increase the level of transcription to produce mRNA. So we can see how everything started right here with the chemical signal binding to the receptor, producing a kind of signaling cascade or internal signal, a passing along of the message, which eventually increased the level of transcription. And that's going to increase the level of translation of the mRNA and the increased production of the protein. So reception is going to be the binding of the chemical signal to the receptor. Transduction is the sending of that signal from the outside to the inside. And response is the end product that we get.